So we just want to quickly look at the importance of mRNA and of the different types of RNA in the transcription translation process, right? So messenger RNA, very, very simple. Messenger RNA is what allows the genetic information that's stored in our nucleus to be accessed by ribosomes and translated into proteins. Without messenger RNA, we would not be able to produce proteins because the ribosomes would not be able to access the information in the DNA, right? Make sense? Good. So a messenger RNA produces a transcript of a gene in transcription. tRNA or transfer RNA, right? They form the bridge between mRNA, the information held in mRNA, and actual polypeptides. Without tRNA, we would not be able to attach specific amino acids based on the genetic information that's held in our mRNA. Remember, the tRNA contains anticodons, which are brought in complementary to the codons on the mRNA, right? And when that takes place, specific amino acids are brought in, and that polypeptide chain is produced. Without tRNA, we would have nothing that reads the sequence on the mRNA and brings in specific amino acids. Does that make sense? It's like a car with no fuel, right? You have the ribosome, but without tRNA, you can't produce an amino acid, a polypeptide. Make sense? So attaches complementary tRNA anticodons to mRNA codons in translation, producing the polypeptide chain, right? Now, ribosomal RNA, this is one you guys haven't heard of before. Ribosomal RNA is basically RNA that is involved in the structure of the ribosome, right? So some of the ribosome, parts of the ribosome are actually made up of RNA, and that RNA is known as ribosomal RNA. So without our rRNA or ribosomal RNA, our ribosomes would not be functional, as bits of its structure would be missing. Make sense? Good. Do we have any questions? No? Okay, I'll give us a break now. Okay, guys. All right. So coming back to this, we're all good with what we've covered so far. Yeah, we don't have anything we're confused about at all. If there's any questions, now's the time to ask. No? Okay, so I will get you guys to do these questions. I'll give you guys some time to do these questions, and then we'll go through the answers together. So everyone, please start working on these questions. In pairs? Do you guys normally do it in pairs? Okay, do it in pairs. Yeah. All right, guys. Okay, so let's. I know you guys are probably working on 2 at the moment. Let's bring our focus back to 1. Right, question 1. Now, I'm assuming we've all finished 1, right? I've given us plenty of time. Yeah. Okay. So, question 1, right? Discuss the key cellular processes required to produce polypeptides from DNA. Use a label diagram in your answer. 6 marks. So this is a good question, particularly now, because it kind of forces you to summarize everything that we've gone through in the first half of the lesson. Now, does anyone want to share their thoughts or what they had? Transcription translation. Okay, good. Now, is transcription translation enough detail? What else do we want to say? RNA processing, okay, good. Some of the things we should be mentioning, this didn't come out. RNA processing, transcription translation, anything else? So what was that? Define the terms, okay, good. tRNA, good. You should definitely talk about tRNA. What else? mRNA? Oh, RNA, yep. So the different types of RNA, mRNA. What about diagrams? What were the diagrams that we had? Well, I mean, you guys had it a bit easy because if you scroll up, there are diagrams up above, right? What do you mean? Can I see it? That's okay. It's not bad. It's thinking outside the box. The what? The diagram. Can you guys hold up the diagram? Okay, that's quite good. Hey, guys, the rest of us? That's good. Some of us in this corner, can I see the diagram? So that's tRNA, I see that. What's the one at the bottom? Transcription. Okay, good. Where's the RNA polymerase? You drew it. Is that in the corner? Okay. Cool. Okay. So guys, for a diagram, right? Yes. That's another thing you should mention. I will go through everything you need to mention. So for our diagrams, for a question like this, since it's the process of getting polypeptides from DNA, right? The two things you should have in your diagram is a diagram of how transcription occurs and then how translation occurs, right? And it doesn't have to be very, very complicated. You can do kind of like you can just draw the strand of DNA, right? Obviously make it neater than this, but draw a strand of DNA, draw an RNA polymerase, and then just have an arrow, right? Like the RNA polymerase can literally just be a blob. And then that produces an mRNA strand, and just draw in some letters and have the sequences, right? So for example, right? Something like that. That's very, very messy. I've done it very, very quickly. Yours should look cleaner than that, but something as simple as that, right? For translation, something similar, right? Have your mRNA strand, have a ribosome. You can draw the different subunits. If you want to go into enough detail to have the three sites, the A, P, and E sites, you can. And then just show the tRNA. Just draw up a quick, it can be a scribble with the amino acid. Show it coming in. Show it bringing in the amino acids. Show the polypeptide chain for them, right? That's all you really need to have. Some of our diagrams are actually quite good, right? Others were okay, but we didn't do a bad job. Okay, now someone online, can you read your answer out, please? Harshul, are you there? That's what? Look, man. Why? Okay, Harshul, are you there? Guys, quiet down, please. Harshul, are you there? Look, man, are you there? Does online students want me to take a while to respond? Huh? I see. Okay, well, the online students are not responding, so I'll bring this back to you guys. Does someone want to read out their answer? I will pick on someone if no one volunteers. Any takers? No. 5, 4, 3, 2. Okay. Can you read out your answer, please? Yeah.
Function of polypeptides consists of two stages, transcription and translation, which are two essential cellular processes responsible for polypeptide synthesis. Transcription refers to the creation of a complementary RNA strand from the antisense strand. This strand is called mRNA and is beneficial as it creates a message containing introns and exons. In order to remove introns, RNA splicing takes place to improve efficiency in translation. To ensure that the newly spliced strand of exons are protected, a 5' count and a poly A tail is added to protect the mRNA from enzymal degradation as it leaves the nucleus. Finally, translation of that mRNA takes place through the complementary matching of codons and anticodons. tRNA contains specific amino acids and anticodons, which matches with the codons to form a chain of amino acids, forming polypeptides and eventually forming peptide bonds and eventually forming polypeptides.
Okay, good. What do we think about his answer? Is there anything we could add or change? No? Good. You covered most of the key processes, and almost hit everything that I would want you guys to talk about, right? Do we all have similar explanations? Yeah. Now, the only things I would suggest you add with transcription and with translation as well, with transcription particularly, just talk about how it starts. Talk about that initiation phase, right? So say there's a specific sequence in the DNA known as a promoter region, right? This is recognized by the RNA polymerase, and that's where transcription starts. The RNA polymerase then moves along the sequence of DNA, creating a complementary strand of mRNA. Okay. Yeah. I am speaking quite fast, so. Yeah. Strand. All right, you guys want some dot points of everything you should cover, more or less. That's very, very in depth, but those are the main points you want to make sure you hit, right? There's some key words in there that you need to hit. I'm sure Prithvi talked to you guys about making sure you include those buzzwords in your answers, right? Because you can explain something, but a student who explains something using the correct keywords and the correct terminology will always have a better answer than a student who explains the exact same thing to the exact same level of depth, but doesn't use those words, right? So obviously mention transcription and translation. I want to mention the promoter region. You want to mention RNA polymerase, complementary mRNA strand, pre-mRNA, RNA processing.
Did you have a question? So you this is discussed provide.
Yeah, I know. So this is a bit of a weird question because with a discuss, right? I'm actually glad you identified that. With a discuss question, I'm sure you guys are like used to seeing discuss and thinking talk about the positives and the negatives, right? But with a question like this, I mean, it's asked you to discuss the key cellular processes, right? With transcription and translation, there's not really any element of positives or negatives, right? Personally, I think this is more of an explained question, but I mean, with discuss, there's not really any way you could logically derive a positive and a negative of transcription and translation. Does that make sense? Yeah. No, I mean you definitely hit the key points. See, the discuss is like the active verb, like discuss. That's to do with how you frame the way you articulate the ideas, but the key ideas that you talk about can be the same. Does that make sense? So even though it's a discuss question, you still need to hit the main points, right? You need to talk about the initiation of transcription. You need to talk about the mRNA strand being complementary to the antisense or the template strand. You need to talk about RNA processing. You need to talk about translation. It's just, for a discuss question, you might talk about it in a slightly different way than you would for an explained question. Does that make sense? All right, this is about that. We have, okay, that's fine. Okay, guys, before I let you guys go, just quickly, I just want to get your ideas on question 2. What are your thoughts on question 2? Good. So compare, so you're directly comparing mRNA and tRNA. So you need to talk about some similarities, some differences, and talk about, obviously, the other keyword, their relative importance, what role they play in polypeptide production. So can someone tell me a similarity between mRNA and tRNA? They're involved in polypeptide production, yes. Just a simpler one. They're single stranded. They're both RNA, right? So both mRNA and tRNA are RNA molecules, right? Or RNA molecules which are involved in the process of polypeptide synthesis, right? What's mRNA? What's the purpose of mRNA? Messenger RNA. Good. Can you explain to me what that means?
You have
Yeah.
you have the DNA strand,
Yeah.
and the mRNA strand is the complementary strand of the DNA strand. So like, I don't know, like.
What's the purpose? What's the purpose? Think purpose, right? Look, it's a bit of a weird analogy, but you can say a car is made of metal, right? But what's the purpose of a car? To get you to places, right? So what's the purpose of the mRNA? Does someone want to help? Good. To convey the genetic information in the DNA, right, to what needs the genetic information, which is the ribosomes, right? So the messenger RNA, the purpose of it is to act as a messenger that takes the information in the DNA and takes it to the ribosomes, right? One of you guys at the back, can you tell me tRNA and its importance?
It helps in producing the polypeptide chain.
Okay, good. Do one of you want to elaborate? Yeah.
It helps with finding.
Good, good, perfect. Do you do something to add?
It has ATAR
Yeah, good. So the mRNA is the first step, right? Taking our genetic information and converting it into a protein. We've gotten the genetic information. We've gotten it out of the nucleus, right? Now we need something that reads those 3 sequences and brings in the respective amino acids, right? And the tRNA is what allows that to happen. The tRNA has anticodons, which are complementary to the codons, the sequences of 3, remember, bases on the mRNA strand, right? And based on the anticodon held by the tRNA, it will carry a specific amino acid, which allows polypeptide production to take place. Good. Any questions? No? We have 2 minutes. So why do we think models, you guys can start packing up, but we'll just talk about this. Why do you think models are necessary to explain such processes? Good. Does anyone else have any ideas? Visual, good. So it's a visual simplification that allows you to better understand how the process is place, right? I can tell you that ribosomes attach, right? There's a small ribosomal subunit and lots of ribosomal subunit. But showing you guys a model helps you better understand how that actually takes place, right? Okay, good. I'll let you guys pack up. If you guys have any questions, feel free to ask. Those of us online, you guys have gone MIA. Can I just get some sort of communication from you guys? Okay.
Hello, can you hear me now?
Yeah. Man, I
there
can hear
you
you.
go.
The rest
yeah,
of sorry, question?
I just want to ask, what is the translational? I saw something about like apparatus.
Okay, so what's the structure that's responsible for carrying out translation?
The structure for translation are ribosomes.
Ribosomes, good. So the translational apparatus is basically just the ribosomes assembling itself onto the mRNA strand.
Oh,
Remember,
right.
there's a small ribosomal subunit and a large ribosomal subunit.
Yep.
It's that process of them attaching to the mRNA.
Yep.
Make sense?
Yeah.
Okay, good. Any other questions? No? All right, I will let you guys go.